she got away.
We lost Dr. Goonfario.
Dr. Goonfario.
Again.
Well, I guess we have to present our case to the force. All right, let's get started. We're doing gene expression, as you can read from the slideshow. Case number 876461, Dr. Goonfario's mission to live forever. All right, let's get into it. All right, here's our table of contents. As you can see, gene selection and amplification. Dr. Goonfario took the FGF2 gene and has started putting it into other human beings as unwilling test subjects, highly illegal, in her mission to live forever. All right, then we have the insertion of the DNA. We're going to go over that so everybody understands what they're looking for and why this is an evil situation. All right, then we have eukaryotic gene expression. It's very important to understand how genes are expressed in eukaryotic cells, such as human cells, which is what we're dealing with today.
Next, we have prokaryotic gene expression, which is important to know because you need to know why Dr. Goonfario used humans as opposed to like the bacteria.
Right. Perfect. Next slide.
Next is how Dr. Goonfario knew his plan worked, how we know he actually did it, which is gel electrophoresis, which is how we see how the DNA has changed.
All right, then we move on to mutation in DNA sequencing. It's important to be able to identify the mutations, all right, so we know where they've come from and why it's happening so we can fix them before they become a bio disaster, all right? And then different types of mutations. We need to know all of them, or else, because if we don't know one, bad things are going to happen, and we're going to go over what they are so you cadets can understand. All right, let's get into it. Oh, I completely forgot. We didn't even
before we start
introduce ourselves. I am Detective Jerry White. All right, I've been on the force for 25 plus years. I am your senior, and I caught the Cincinnati cloner. All right, Dr. Goonfario is next. This is my partner.
My name's Karen Doofenshmirtz. I'm a forensic scientist with master's in criminology and biology from Tabony University.
The best university. I didn't go there myself, but I've heard good things. All right, now let's get into the evil scientific mastermind that we are hunting today. Also, before we get into that, actually, we are of the FBI, Forensic Biology Investigators. Okay, we handle all biology-related crimes. All right, now let's take a look at our criminal for today. Dr. Goonfario, a highly ranked scientist, also a graduate of Tabony University, but took the expert education she learned there and decided to use it for evil. Wants to live forever, so she can learn how to recreate Fortnite in real life. I've never heard of anything more evil than that. All right, and doesn't know how to cook. Why is this on the slide? Irrelevant. And hates anyone who has a PhD in biology. That would be us. That's why we're nemesis. All right. Failed biology 6 times. Isn't that crazy? How are you going to be an evil scientist and fail biology 6 times? At least be good at your job. Needs to be stopped. That's why horrific things are happening. Let's get into our first round of background evidence. Gene selection and amplification. We're going to go over what gene was selected, the primers that were used, and how to work a PCR, because you need to know all that information if you're going to catch Dr. Goonfario. All right, Karen, I can take it from here.
Okay.
Karen will be back, trust. All right, gene selection and amplification. Step 1 in Dr. Goonfario's plot was choosing the gene. The doctor chose the FGF2 gene because it aids in regeneration and the healing of cells, which I don't know if you know this, but rapidly healing cells lead to fast wound healing and all of those other things that helps with the prolonging of a life, which is Dr. Goonfario's mission. Then he had to identify primers, a forward primer and a reverse primer. All right, that's how you're going to start the copying process of the gene. Then these primers are going to attach the beginning and the end of the gene so the copying happens in the right place. This is important. Remember that. Okay, let's talk about setting up the PCR in its formal name, the polymerase chain reaction. Okay, so here's what we need in a PCR test tube. We need DNA sample, the forward and reverse primers we identified previously, DNA building blocks, Taq polymerase, buffer and magnesium, and water. So it all has the same correct volume. All right, are we following? Good. Next slide, please. All right, step 3 of the PCR machine. The PCR machine heats and cools a mixture and cycles to copy DNA. It follows the following steps. Memorize this. Write this down in your notebooks. All right. Denaturation, annealing, extension, and then those are the 3 steps, and then you repeat it 30 to 35 times, which seems like a lot, but it's really not. Okay, so don't get all lazy on me. Then we have to check if it worked, and we use gel electrophoresis. Okay, and we're going to get into that later, so if you feel like you're missing out, don't worry. We have a whole slide on it. later on in the presentation. Okay, on to the next slide. Insertion of the DNA. Now we're going to go over how the DNA is inserted into the human cells, okay? This is, I would say, a very important part of Dr. Goonfario's plot. It is the one that took him the longest. All right, we are going to use CRISPR-Cas9, which is the substance we're going to use to cut out the DNA and then reinsert it in another cell. Okay, so we need a guide RNA. This tells the Cas9 exactly where to cut, so no mistakes are made, okay? Then the Cas9 enzyme acts like a pair of scissors, cuts out the DNA, and then places it somewhere else, okay? The donor DNA of the FGF2 gene, and then this is the new gene we want to insert into the other cell, okay? Then we need a target site in the human genome. Now, a sensible person would use a safe harbor locus like the AABS1 chromosome or site on chromosome 19, which allows staple gene insertion, but not Dr. Goonfario, okay? That evil scientist is using harmful sites on chromosomes like chromosome 1, 2, the ones that really affect your development and gene expression, okay? Be careful. Then, step 3, editing the human genome with CRISPR-Cas9. All right, so the Cas9 and the gRNA find the target DNA, okay? They find it, and the gRNA directs the Cas9 to the chosen site on the genome, all right? Then, the Cas9 cuts the DNA like a nice little pair of scissors and makes a double-strand break, okay? Then the cells try to repair the broken strand using the donor DNA that we gave it previously, okay? Then the FGF2 gene pastes itself into the homology-directed repair, HDR. So basically what is happening is that the Cas9 is triggering the healing response in the cell, and then it's using the DNA that Dr. Goonfario placed in it to repair itself, therefore editing the DNA of the cell itself. Isn't that some diabolical science? All right. Oh. Then we got to check if it worked. All right, we have to confirm that the CRISPR edits were inserted by PCR, which we already went through. That's how we figured out if it worked. And then a Western blot. This is not as relevant. And then here's our summary. The guide RNA and the Cas9 cut the DNA at a safe spot, or if you're Dr. Goonfario, at a not safe spot because he doesn't care about the well-being of his involuntary test subjects. Getting worked up. All right. Then the FGF2 gene is inserted using the cell repair system, which we went over previously. Then cells are tested to ensure successful editing. All right. Eukaryotic gene expression. Let's get into it. I'm going to do the first couple points, and then my partner is going to take over. All right. DNA. So the first thing is DNA access, chromatin remodeling. It's pretty much what it sounds like. All right. So the DNA in eukaryotic cells is wrapped around proteins, okay? And they're called histones, forming chromatin, which is important into the DNA. All right. Then the tightly packed chromatin are inaccessible and not expressed, okay, because they're so tightly packed together. Okay, then there are loosely packed chromatin, which are accessible and can be transcribed. Okay, so then the modification of the histamines or DNA control access are acetylation, which loosens the chromatones, allowing them to be accessed and expressed, and methylation, which often silences genes by tightening chromatin. Okay, next slide. Then we have the transcription, the DNA to the pre-mRNA. So, regulation levels, pre-transcription control. This step occurs in the nucleus, which is controlled by transcription factors. So, let's talk about the key steps of this part. Transcription factors. Okay, they bind the promoter region to the Tata box. Funny name, by the way. Then the RNA polymerase binds the promoter and starts copying the gene into pre-RNA, which is going to help the eukaryotic gene expression process. Okay, then there are enhancers and silencers, which we talked about earlier with the chromatin, which was acetylation and methylation. Okay, so the enhancers increase the transcription and the silencers suppress transcription. This is a very useful cell mechanism, which helps make sure the correct gene is expressed at the right time. And then mRNA is synthesis in the 5-3 direction. Very important. Okay. Now, my partner.
Okay, post-transcriptional regulation. The newly made pre-RNA must be processed before leaving the nucleus. First, a 5' cap is added. A modified guanine cap is added to the 5' end to protect the mRNA and help with ribosome binding. Poly-A tail addition. A long chain of adenines poly-A tail is added to the 3' end to protect mRNA from degradation. And the last step, splicing. Introns, non-coding regions are removed, exons, coding regions, are spliced together. Alternative splicing can create different proteins from the same gene.
Oh, I'll take this one. All right, now we're going to talk about mRNA transport into the cytoplasm. So this step is pretty cut and dry, pretty easy. It's basically when the process mRNA exits the nucleus through the nuclear pore and into the cytoplasm where translation occurs. And then there's some regulation that happens so things don't get crazy. Okay, some RNAs may be degraded or stored before translation. So it's not, they don't always go through all at the same time. All right, translation, RNA into polypeptide. So, my partner again.
I'll take this one. Pre-translation. The processing steps that occur on a newly transcribed RNA molecule in eukaryotic cells before it can be used as a template for protein synthesis. Some of the key points are pre-RNA. The initial RNA transcript produced by DNA transcription in eukaryotes is considered pre-RNA, mRNA, and needs further processing before it can be translated into a protein. Second, splicing. Non-coding regions, introns, are removed from the pre-mRNA, and the remaining coding regions, exons, are joined together to form the mature mRNA. Third is the 5' cap and poly A tail. Additional modifications include adding a special nucleotide cap at the end of the 5' end and a string of adenine nucleotides, poly A tail, at the end of the 3' end and the pre-mRNA. Fourth, regulation and gene expression. Pre-translation processing provides a crucial point for regulating genes expression by controlling which parts of the pre-mRNA are spliced out, potentially leading to a different protein variants from a single gene.
All right, step 6, the last step of eukaryotic gene expression. So we have post-translation modifications, protein processing, and regulation. Okay, so we have regulation level, post-translation control. So the newly made protein must be folded and modified before it becomes functional. Very important. Then the modifications include folding. They must fold into the correct shape. The proteins must fold into correct shapes. Then we have cleavage. Some proteins are cut to become active. Okay. Then we have the addition of chemical groups, phosphorylation, glycosylation, and ubiquitination. Lastly, we have the transport. The proteins are sent to different parts of the cells or secreted outside via the Golgi apparatus. And that is eukaryotic cell expression.
Prokaryotic gene expression. You might be wondering, what's the difference between eukaryotic and prokaryotic cells when they express their genes? Well, in prokaryotic cells, it happens in the cytoplasm because of the lack of they don't have a nucleus like eukaryotic cells. Second, they regulate their gene expression and metabolism through regulating their transcription. Transcription and translation both happen simultaneously in the same place. This makes it a lot more efficient than eukaryotic cells. Prokaryotic gene expression is also a lot simpler than gene expression in eukaryotes. Next is operons, which are only found in prokaryotic cells. And they're organized clusters of genes that all contribute to a particular metabolic task. There are two types, inducible and repressible operons. Inducible are for metabolic pathways that are usually turned off. It controls gene expression in response to a stimulus. For example, in this one, it's normally turned off, and so it's waiting for a stimulus to turn it on. And repressible is a group of genes that are usually on but can be turned off by a repressor protein. For example, there's a gene, and it's turned on. But if it needs to be turned off, it's waiting for a repressor protein to turn it off. Why did Dr. Goonfario use eukaryotic cells? Well, eukaryotic cells are much more complex than prokaryotic cells and are closer to human cells, which are also eukaryotic. And it is important to study the complex systems of multicellular organisms, which you cannot find in prokaryotic cells. Next, in order to prove that he actually went through with his science, we need to do gel electrophoresis to prove it. But before we do gel electrophoresis, we must isolate the DNA and digest using restriction enzymes. Restriction enzymes recognize palindromic DNA sequences and cut in the middle of the sequence and leave a 5' overhang and a few bases. These are called sticky ends. These two right here. This allowed us to select the sequence he edited to run through gel electrophoresis in order to compare to regular DNA. It's gel electrophoresis time. The steps to run a gel are, first, you have to prepare your samples. Second, you have to prepare your machine by putting the agar sheet in correctly in the water, matching up the red and the black. Third, load samples into wells right here. And then run the gel in the machine. And then after that is done, you need to observe the banding on the gel. So now that we have run the gel, we need to analyze the results. The location and the thickness of the bands are important markers for the DNA to tell us what happened. The thicker the band is, the more DNA is present in that band. So in conclusion, Dr. Goonfario's experiment succeeded because all these bands are thicker in the area of DNA of cell regeneration, which will cause reduced cell death and allowing organisms to live forever.
All right. Let's take a 5-minute break from our case presentation.
Yeah.
I'm saying we hit the bathroom.
Yeah.
All right. We'll be back.
Wait, should we lock our office?
Nah, it's probably fine. So how's the kids?
Oh, they're good. They're crazy.
They always are.
Yeah.
That's what I'm saying.
You know, my wife Cheryl, she's always harassing me for another kid. And I'm like, we already have 2. Oh, Dr. Goonfario! Oh my god, he locked us in. Oh, he locked it. Open the door.
It is I, Dr. Goonfario, and I have taken over. This is now my slideshow. So I'm going to be talking about mutation and DNA sequencing. My experiment is working and everyone is able to live forever. So I'm going to do this on more people. Uh-oh. Something went wrong. I think it's a mutation. How can we compare the original and suspected mutant strain to verify the mutation? DNA sequencing. Okay, so the steps to DNA sequencing. We've talked about this before, but first, PCR. We have to run a PCR so we can create multiple copies of the DNA that I think is mutated. Then we're going to add deoxynucleotides. The ddNTPs can terminate the elongation step of PCR once they are incorporated into DNA. Fluorescence is also added to the ddNTPs so that it is easier to differentiate between the bases. Then we're going to go back to gel electrophoresis. This is where the fragments are run through. Short fragments move more quickly and long fragments move slowly. And lastly, we'll perform a computer analysis so that we can compare the 2 DNA sequences, the original DNA and the mutant one. Types of mutations. So there are 2 major types of DNA level mutations, point mutations and frameshift mutations. In point mutations, one DNA base is replaced by another DNA base. And then in frameshift mutations, DNA bases are inserted or deleted, or indel. So, types of mutations are silent, missense, and nonsense. A silent mutation is when the substitution change in a codon to another codon for the same amino acid. So, like this picture here. Then, a missense mutation is the substitution changes a codon to a codon for a different amino acid, like this picture here. And then a nonsense mutation is when the substitution changes a codon to a stop codon, like this one here, right here. Okay, and then types of frameshift mutations include extensive missense, immediate nonsense, and limited effect. Extensive missense means the reading frame of the ribosome is altered so that all amino acids downstream from the indel are altered, like this. And then in immediate nonsense, the reading frame of the ribosome is altered so that a stop codon is introduced prematurely, right here. In limited effect mutations, the reading frame is restored when indels occur in multiples of 3, like this. They'll never catch me now, because now I can know which mutation it is and I will fix it.
Hurry up, old man. I'm trying. It's harder than it looks. I got it. Took you long enough. I got it. Dr. Goonfario, right freeze!
No, you'll never catch me.
Dr. Goonfario, she got away again.
Got away.
All right, cadets, this isn't over. You still have a test on Monday. All right.
Study up. Study up.
Study up, okay? And don't you worry. This isn't over yet. Just be ready. Stay aware. We have a doctor. We have an evil scientist to catch. See you next time.
Three, two, one, go. Oh. Okay, go.
Wait,
Oh.
do another countdown.
Okay.
Yeah, those dang kids. I'll tell you.
Mhm.
I have two, and they're such a handful. You know, my wife Cheryl, she's always asking me for another kid, and I'm like, we already have two. Crazy, you know. What was that? What was that? Oh, I did Dr. Goonfario! Testing. Audio quality. Testing again. Testing to see if moving the mic works.
Karen Doofenshmirtz.
And Jerry Watt.
Oh, oops. I just took a screenshot and a video.